We're going to continue our conversation about digestion with proteins. So our next step will be proteins. But before we can really talk about digesting proteins, we need to talk about the structure. So remember with proteins, they're really just a chain of amino acids. We have an amino group on one end, a carboxylic acid on the other end, because really what it is a chain of amino acids making up our polypeptide or making up our polymer. So when we break down proteins, we're doing it in a catabolic mannerism, catabolism, which remember means we're breaking large molecules into smaller molecules. So ultimately, what we're going to do through a few steps is break these polymers into the individual monomers, the amino acids. So there'll be some steps to do that. And like you noticed with the carbohydrates, we start with the large molecule, go a little smaller, a little smaller, a little smaller, until we eventually get down to the individual units. We're going to do the breakdown of the proteins in a manner very similar to what we did with carbohydrates. We're going to break the peptide bonds. So of course, the peptide bonds are characteristic of the proteins, but we're going to break them by adding water, by doing a hydrolytic reaction. So remember, lysis is splitting. We're splitting the water, we're splitting the other molecule, putting an H on one side, OH on the other, the hydrogen and the hydroxyl from the water to do our split. So you'll see us progressing through that as we go through our story. So our story is going to start in the stomach. We don't actually do much in the mouth, esophagus, pharynx, that area. So our story will really start in the stomach for proteins. So we've got a couple of different cells in the stomach that do a lot of the work. So we're going to say we were, I don't know, eating a ham sandwich. We had some nice thick slices of ham in there. So lots of protein. So we ate the ham. Of course, we chewed it in the mouth, passed it to the stomach, but that's where the real work begins. So the first cell or type of cell we're going to talk about is our chief cells. So remember the chief cells. We said when we first talked about them, the chief drinks Pepsi. And what we meant by that is it was kind of a goofy way of remembering that the chief cells make pepsinogen. Gen means it'll generate something else, in this case, pepsin. So this is the inactive form. So we also have the parietal cells. So we'll put some parietal cells in here. We have parietal cells, and they make something else that we need too. The parietal cells are going to make the hydrochloric acid. So the hydrochloric acid is what we often think of as our stomach acid. That's what brings the pH of the stomach down to that 1.8 to about 3.5. And that's the pH neighborhood that the pepsinogen likes. So what's going to happen when we're down in that pH neighborhood is the pepsinogen is going to get converted to pepsin. So the pepsinogen was our inactive form. The hydrochloric acid is basically activating it to pepsin. So we'll activate it to pepsin. But one of the interesting parts of that is we have kind of a loop going on with it. Once we get the pepsin, pepsin will go back, activate more pepsinogen to make more pepsin. So the hydrochloric acid starts the process, but pepsin can continue it and keep us making more pepsin. As long as the pH stays below that 3.5, we'll keep going. If the pH ends up getting greater than 3.5, then we'll go the other way. The pepsin will go back to being pepsinogen. So we'll inactivate it by the pH going back to a pH higher than 3.5. So those are the cells in the stomach that start doing the work for us. So what pepsin is going to do is start with that big polypeptide molecule. So I have my big protein. And what the pepsin's going to do is start breaking it down into smaller polypeptide molecules. So you'll notice a continuing theme of going smaller and smaller and smaller. So we'll take that large polypeptide, we'll go down into some smaller ones. So that's what we have happening in the stomach. Then we're going to move on into the small intestine. So we'll start the story in the duodenum. First part of the small intestine, the first foot or so of the small intestine. So the peristalsis sends the chyme, the proteins mixed with the stomach contents, into the first foot of the small intestine, which is the duodenum. So we have different mucosal cells in there, different types of mucosal cells that will look and see, is that chyme high in protein, high in carbs, high in protons, high in fats? And what happens, depending on what we see in there, what the different mucosal cells see in there, will release different hormones. So our story is going to continue in a story about enzymes as well as a story about hormones. So some of the hormones will release cholecystokinin. We talked about it. We might release some secretin. We might release some GIP. And we'll talk about that a little bit later, glucose-synucleotropic peptide. So we'll come back and talk about that. So what we're going to do is we're going to keep talking about what do we do with these smaller proteins now? How do we continue that story? So I think we'll get a new slide and get more room to do it. So we're going to say this is the small intestine, and we're going to keep traveling along that small intestine. We'll have some places where things can enter the small intestine. And we'll talk about what the different hormones and enzymes do. So what we have coming in right now is we have those smaller polypeptides coming in. We're going to have some enzymes that can act on those. And we'll have some hormones causing the release of those enzymes. So the pancreas can release some proteases. The cholecystokinin goes to the pancreas, stimulates the pancreas to release the proteases. So some of the enzymes that we are going to release, the one that primarily gets released is trypsinogen. And again, gen on the end of it means an inactive form. We're going to use an enterokinase. Convert that to trypsin, the active form. So same idea as I did with pepsinogen and trypsin, or pepsin, I'm sorry. Trypsin, once we have it, can activate more trypsinogen, make more trypsin. So we get that feedback loop going on there. But that's not the only feedback loop. We have some other enzymes that we're going to get released. We're going to release chymotrypsinogen. We're going to release procarboxypeptidase. We're also going to release proelastase. So we've got 3 pro-something enzymes here, which pro-something means we're releasing inactive forms. But trypsin will kick in and activate them. So trypsin is going to activate chymotrypsinogen to chymotrypsin. Trypsin is going to activate procarboxypeptidase to carboxypeptidase, active form. Trypsin is going to activate proelastase to elastase. So the active form. So we now have these active enzymes and we're ready to start looking at what they're going to break down. So that will be our next step. So we'll do a new slide for that. So we have 4 active enzymes right now. So what we'll do is we'll go through the 4 of them and talk about what they're going to break, how they're going to break it. So my first one is trypsin. Second is my chymotrypsin. Carboxypeptidase. And elastase. So we'll go through them one by one. So first one, the trypsin, it will break at the lysine and the arginine on the carboxy side of a small polypeptide. So this one likes to break the small polypeptide either lysine or arginine on the carboxy side. And as a result, we're going to get 2 things. We're going to get tripeptides, 3 amino acids in a chain, and we're going to get dipeptides, 2 amino acids in a chain or 2 connected. Next one is chymotrypsin. So let's look at a structure for a second because of where that breaks. So this one's going to break the small polypeptide at one of the aromatics. It's going to break at the tryptophan, going to break at a tyrosine, or going to break at a phenylalanine. I guess since we talked about the structure, we probably don't have to go back and do those structures. But when we do break there, we're going to get the same 2 things. We're going to get tripeptides, and we're going to get dipeptides. Then we have 2 more. They actually do the same thing. The carboxypeptidase and the elastase do the same thing. I guess we're not going to get to change colors here. Okay, that's all right. So what they're going to do is they're going to attack the carboxy end, as the name would imply, of our small polypeptide. They're going to break, and what I mean by that is we're going to break off the amino acids from the carboxy end. Oops. And what we're going to get, going to sound kind of familiar. We're going to get tripeptides, 3 in the chain, dipeptides, 2 attached, and we're going to get some free amino acids now with this one. So what do we have now? We have tripeptides, we have dipeptides, and we have amino acids. So now we can keep moving through the small intestine, through the rest of the small intestine, and we can talk about what's going to happen at the enterocytes. So you talked about enterocytes in histology. So we're going to talk about them a little bit more. So we have our enterocyte. We're going to make a nice big enterocyte so we have room to draw in it. So this will be our enterocyte. And remember that enterocyte has the microvilli on the membrane, have the brush border enzymes, have lots of stuff in there. So we'll draw that too. So we have our enterocyte. We have our microvilli. And we have brush border enzymes in the microvilli. I did kind of a terrible job drawing the microvilli, but what we have is a nice high surface area. So we can have lots of enzymes, lots of molecules embedded in that. Remember we said if you see a membrane or anything with a lot of wrinkles, the wrinkles are usually about having lots of surface area. Lots of surface area is about either having lots of stuff, room to have lots of stuff embedded in it, and or lots of room to pass molecules and other things through it. So we have the brush border enzymes. Embedded, and we'll talk about what they are and what they're going to do. We have, in our case right now, we have 2 types, and the names are going to be pretty descriptive, which enzyme names typically are. We're going to talk about our amino peptidases, and we're going to talk about our dipeptidases. And remember early on when we talked about terminology a little bit, we said that if you see am or pep as part of the word, you're probably going to be in the neighborhood of peptides, proteins, amino acids, that kind of thing. We said GLY, GL anything. Think about carbs. Acyl, acyl, ester, lip, anything. You're going to start thinking about fats at that point. So anyway, peptidases, things like that. Amino, you're definitely in the protein neighborhood. So we're going to draw a couple of enzymes on here. This will be my dipeptidase. We'll put some transporters in here. We'll have an amino peptidase since we were talking about it. We need that. And we'll talk about what they're going to do. So right now we have 3 kinds of molecules. We have lots of molecules, but some of them are tripeptides. Some of them are dipeptides. Some of them are single free amino acids. So we still have peptide bonds to break. So we're going to do a couple of things, and you can kind of, it's pretty intuitive on where we're going here. So we'll start out, we'll take since we have dipeptidase on the top, we'll talk about the dipeptides. They're going to go to the dipeptidase, and we're going to break that bond and get free amino acids from that. So we'll break the dipeptide into free amino acids. We can go to the aminopeptidase with a couple of different things. We'll take the tripeptides there. So we'll take the tripeptides, we can go to the aminopeptidase there. What that's going to do, the aminopeptidase will start breaking the peptide bonds, but it'll start at the amino end. So remember, we had our amino acid, we had our amino group, but the end made at the N terminus. We had our carboxylic acid, the C made at the C terminus. So this enzyme starts cleaving at the amino end, starts breaking the peptide bonds. So what it will do is it'll come in, break a bond, break a bond. So what it'll give us, let me go back to blue for amino acids, is it's going to give us some free amino acids. So we're going to get a lot of the tris and dis broken down into free amino acids, but we're going to still have some dis and some tris left. So we have 3 transporters, really simple on the transporters. We have a transporter for the tripeptides. We have a transporter for dipeptides. We have, you guessed it, a transporter for the amino acids. So what we're going to have happen is everything is going to go in through its transporter. So what we're going to end up with is all of these things in the enterocyte. We'll probably have to draw another enterocyte. We're going to run out of room here. And we've got a couple of other things going on too, helping these transporters transport. So the tris are going to go through the transporter for the tripeptides, dis for the dis, free amino acids for the free amino acids. But we've got some help here. And we've got some help for a couple of them, the dipeptides and the tripeptides. So the help we need there, well, we need help for all of them, but we'll talk about the helper for the tris and dis first. So what we're going to do is we're going to set up kind of a concentration gradient. We're going to use a cotransporter or a SIM mechanism. We're going to use a sodium hydrogen ion transporter. And what this does is we're always going to have sodium going into the cell. We're going to have hydrogen coming out of the cell. So sodium's going in, hydrogen's coming out. So as we're doing that, we're going to use that hydrogen coming back in to help the tris and the dis come in. So this is our cotransport or SIM mechanism. These hydrogens are going to come back in with the tripeptides. They're going to come back in with the dipeptides. So that's one of our cotransport mechanisms going on. The amino acids are going against the concentration gradient. The sodium's going with the concentration gradient. So we're kind of setting that up. Then when we want to bring the individual amino acids in, we've got a little bit of a story there too. We have another transporter, and this is going on in every cell. We've always got sodiums going out. We've always got potassiums coming in. So We're always doing this. So the sodium concentration in the cell is always decreasing because the sodiums are always leaving. So since the sodiums are always leaving, they're outside the cell, the amino acids are going against the concentration gradient. If the sodium kind of helps them come in, since it's going with the concentration gradient, that will help get the sodiums, or the sodiums will help get the amino acids into the cell, if that makes sense. And I know these transporters is something you talk about in other physiology classes too. So what we have going on now is we have amino acids in there, we have dipeptides in there, we have tripeptides in there, and we're in the enterocyte now. So what we're going to do is we're going to use peptidase, break the tripeptides down into amino acids. We're going to use peptidase, break the dipeptides down into amino acids. So now we have all these individual amino acids. So we have one more thing we're going to do. We're going to take them, and we're going to send them out. So we're going to use facilitated diffusion to transport the amino acids out of the cell. They're going to go into the bloodstream. So our carbohydrates went into the bloodstream, our amino acids go into the bloodstream. They're going to go through the hepatic portal vein, can go to the liver, and we'll continue that story from there. So that's our story of getting our proteins into the bloodstream. Next story we're going to go to is getting lipids into the bloodstream.